Next up, we have Jody Bonnevier from Bio-Techne, monitoring the interaction of SARS-CoV-2 with ACE2 and the resulting immune response.
Great, thanks so much. Good afternoon, and thank you for joining my presentation today. Thank you, Tina, Kathy, and the organizers for this opportunity to share some really exciting new data with you today. I manage a new product development group in the antibody business unit at R&D Systems Bio-Techne at our headquarters in Minneapolis. So R&D Systems is part of the Bio-Techne family. Bio-Techne brings together the prestigious life science research brands of R&D Systems, NOVUS Biologicals, supplier of 200,000 plus antibodies, Tocris, a leading supplier of small molecules for cell activation and inhibition, Protein Simple, developer of proprietary instrumentation, including WES, an automated gel-free Western blot platform, and ACD, Advanced Cell Diagnostics, a leader in molecular pathology with RNAscope for in situ hybridization and the ability to co-localize RNA and protein with IHC-ish. Bio-Techne has been developing and manufacturing antibodies for over 30 years. All products are manufactured under compliance with ISO guidelines, and certain products are FDA cleared or manufactured to GMP grade. Our antibodies are well suited for flow cytometry because many are generated from whole protein, high affinity to natural conformation proteins. Because of this deep pipeline and the antibody development and validation capabilities of our teams, we were able to quickly pivot resources to address the research and pharma community's needs for high-quality ACE2 antibodies for use in all applications, but particularly for flow cytometry. So ACE2, a term I'm sure everyone hears every day, every hour these days, angiotensin converting enzyme 2 is a widely expressed receptor that is hijacked by coronaviruses and used as a port of entry into cells. The expression of ability to viral infection and ways to block viral entry to cells, such as use of an ACE2, an anti-ACE2 antibody, as I'm showing in this cartoon here. Many of the mild coronaviruses, such as common colds, can infect the upper respiratory tract, and which is make them quite infectious. And in contrast, SARS-CoV and the MERS virus were known to infect the lung, not so much the upper respiratory, but the lung, and that made them quite deadly, but not quite as infectious. Unfortunately, SARS-CoV-2 seems to have the ability to do both, making it a double whammy. So here I'm showing you some IHC staining using an R&D Systems MAB 933. So obviously ACE is in the lung, so this is some alveoli staining, showing some nice staining there. ACE2, as I said, is in quite a few other tissues. So one of the other potential points of entry is in the small intestine. This is some staining that I took from the Human Protein Atlas, where this antibody has been extensively characterized. And then there's also staining in kidney, there's staining in blood vessels, and a number of other tissues. So while we're learning a lot about ACE2 expression in tissues from these types of IHC staining, its expression in other cells like circulating blood cells that's still a little bit unknown and really an area of intense research. And one of the reasons we don't know too much about where ACE2 might be expressed on lymphocytes or myeloid cells is that there hasn't really been a robust ACE2 antibody available for flow cytometry studies. So as I mentioned, we have a very deep antibody development pipeline, and while we pick one or maybe two really highly characterized and validated antibodies from a hybridoma fusion panel to commercialize, we usually have many more of what we call sister clones from that fusion panel that are in our freezers. And those antibodies are not commercialized, but they're just, you know, kind of available. So we have those available at our disposal in our development teams to very quickly pick up and test in all kinds of different ways. They're there at our disposal when we need them for a new need. They're also available for you as researchers to pick up and buy and test for what you need in any kind of applications. So know that those are available too for whatever you may need. So because of that, we were able to quickly pick up and test a bunch of antibodies. So for human ACE2, we probably have between R&D Systems and Novus, we probably have at least 40 different antibodies available to ACE2. So we've got the luxury of having a large number of antibodies available to test and really pick out the ones that we're happy with for flow cytometry applications. So what I'm showing you here is some brand new data. And one of the ways that we sometimes test flow cytometry antibodies when we don't have a good handle on which circulating blood cells might be expressing a target of interest, we use an overexpression system of some transfectant cells that we generate in-house. So these cells are overexpressing human ACE2 in the, along with EGFP. And then we can use these cells to as kind of a model for the ACE2 antibody. So in the top panel, I'm showing you these cells. The ACE2 antibody is staining the cells in along with the EGFP. In the bottom panel, I'm showing you some specificity, and this is really important in our antibody validation process. So these cells are, they're the same kind of cells. They're HEC293 cells expressing EGFP along with overexpression of some irrelevant protein. And this is really important because we need to make sure that the antibody is specific for ACE2, not something else on the HEC293 cells, not some other protein. So we do a lot of testing on these irrelevant transfectants. So this is a new clone that was just available last week. So that's on the market now, ready for testing, both as unconjugated and in a number of different fluorochrome conjugations. So in addition to that new clone, we have also recently just added the flow cytometry application to a goat polyclonal. The catalog number is AF933. And what I'm showing you here is some data on, again, the same cell model, the EGFP overexpressing ACE2 transfectants. And in this case, the polyclonal antibody binds to human ACE2 transfectants. And then we are also able to show that it does bind to mouse ACE2 transfectants. So it's cross-reactive with mouse ACE2. And then also pretty exciting for us to see that it was also cross-reactive with hamster ACE2 on transfectants. And we were happy to see that because hamster is a widely used model in infectious disease models. So it was great to see that we have an antibody that was able to bind hamster ACE2. So we've had a lot of requests for this antibody. This is an existing antibody. As I said, we just added the flow cytometry application to it. And when we do that, we also add the direct conjugates at the same time. So we've had a lot of customer requests for direct conjugates of this antibody. So those are also just available this week. So those are also available for your use. You may be asking, goat polyclonal, that is probably not my first choice for flow cytometry, and I would absolutely agree with you on that. A polyclonal is probably not my first choice either, but this antibody is been validated, tested, published quite a bit, and when we do have an antibody that has been extensively tested, I feel more comfortable in recommending it for flow cytometry. So let me show you a little bit of that data. This was just a recent publication in Cell from Hoffman et al. And this was a really interesting paper in which they showed that this polyclonal antibody to ACE2 was able to neutralize the SARS-CoV-2 entry into cells. So in this system, they used a pseudovirion, and they used a system with that the viro cells. So they showed that the antibody was able to block that entry of that pseudovirion, and they used both SARS and SARS-CoV-2, and they showed that that was able to block entry into the cells. So that is a pretty exciting finding, and it's the ability of this antibody to neutralize further allows us to characterize that antibody. So we know that a lot of our customers would like to do some type of neutralization or blocking assay, but obviously this type of assay requires a lot of capabilities for a BSL-3 lab. The ability to make a pseudovirion, that's not something that everybody has at their disposal. So we set out to develop a blocking assay that was something that other people would be able to do, something that we could do using a recombinant protein system. So let me show you what that assay looks like. Again, I'm using the same cells. So these are ACE2 transfectants. Again, they're EGFP positive so that we can see the cells. And the cells express ACE2. And the model that we're developing here is a recombinant SARS-CoV-2 spike RBD protein binding to the ACE2. That protein has either a His tag or an Fc tag, and that tag is then detected with an anti-His or anti-Fc secondary antibody, which is conjugated to fluorochrome. These proteins that I'm going to talk about and show you some data for today, these recombinant proteins are brand new proteins that R&D Systems has just released. And in the process of our development of this assay, we have used dozens and dozens of different proteins available from many different vendors. And we've done a lot of comparison of different proteins. I'll show you a little bit of comparison data, but know that this RBD protein really does work very well in this assay. So what you can see when the protein binds, you can obviously see the EGFP colocalized with the protein. And then what this does is it allows you to set up this assay. If you have an antibody or a small molecule or a protein or any other reagent that you want to test to see if it can interrupt this interaction, that gives you a nice robust assay to be able to do that. So here's some titration data. So this is the R&D Systems protein that I was just talking about. So these are all SARS-CoV-2 spike protein RBDs, His tagged. The new R&D Systems protein is in the center, and then two different competitor proteins are on each side. And this is just a titration to show that we really evaluate very carefully the binding of the protein to ACE2 and make sure that it performs exactly as we want it to in this assay. And here's some real data for you. So this is the SARS-CoV-2 spike RBD protein binding to the ACE2 transfectant. So you can see in the dot plot, so the GFP and the protein are colocalized in the upper right quadrant. So we've obviously run a lot of our ACE2 antibodies through this assay to try to find a blocking antibody. And I'm going to show you some data here. I'm going to introduce you to one of the antibodies that does block. So this is a NOVUS catalog antibody, and it's NBP280038. And you can see when that antibody is added, it disrupts that interaction, and then you lose that staining in the upper right quadrant. So this just provides a really robust assay for any type of interruption of receptor ligand binding. This is similar data, and it just shows you just a little bit more specificity. So in just a little bit different way than the dot plot. So the dotted black line is the binding of the spike S1 RBD to the ACE2 cells. When you add the the anti-ACE2 antibody, you disrupt that binding and you block that. So that's the orange histogram there. And in this case, I'm just adding one more piece, and that is an irrelevant antibody. So the blue line is a DC sign antibody, showing that if you add an antibody that does not disrupt that interaction, you still see the binding, and that just adds another layer of specificity, so that you're sure that the assay is robust and specific. All right, I will just add a quick note there that this assay is something that is available as a custom service. We can absolutely run this assay for you. And that's something, if you're interested in, just go ahead and contact our custom service team, and they would be more than happy to start a conversation with you about any needs that you might have with that. So obviously what we would like to know is where is ACE2? So one of the things that we've been focusing on is the expression of ACE2 in the myeloid population. So we've done quite a bit of work recently in characterizing different macrophage subsets. So we've worked out quite a few different protocols for differentiation of M1 and M2A macrophages in culture. So these are using different serums, FBS or human serum, and then different polarizing cytokines, and looking at a number of different markers. I know this is super small, but I just wanted to give you an overview of kind of some different macrophage markers subsetting that's available, and a link to this on our website if you wanted to dig into some more of the details about any of these markers. Unfortunately, I don't have any exciting ACE2 data for you yet today, but this is an active area of research in my team right now. So one of the unfortunate outcomes of COVID right now is the cytokine storm that kind of takes over as patients are undergoing this massive immune response trying to fight the virus. And one of the reagents that is in testing right now as a potential therapy is SARILUMAB. This is a blocking antibody to the IL-6 receptor. So we do have a biosimilar available for any type of in vitro testing that you might be interested in for looking at this interaction between IL-6 and the IL-6 receptor. So this is similar to the ACE blocking study that I just showed you. We do obviously do this blocking study for a number of different ligand receptor interactions. This one I'm showing you is on U937 cells, although we have done this too on IL-6 receptor alpha transfectants, and we can see the same thing. So the U937 cells express the IL-6 receptor alpha. The ligand in this case is IL-6 biotin protein. This is also a product in our catalog, and then the detection is with streptavidin PE. So you can see the colors are a little bit different on this one, but the binding of the IL-6 biotin protein with streptavidin PE is in the orange histogram. When you add the SARILUMAB biosimilar, that interaction is disrupted down to the level of just adding the streptavidin PE alone. Right, and I see that I have talked very fast, but I'll just go through a couple of acknowledgements here. So I'll just acknowledge the people in my group that did all of this work and that put together all of this great data. So Christine Gatz, Christopher Hammerbeck, Brian Peterson, Crystal Newman, Lee Pang, and Jay Hugh in my team did all of the flow cytometry work and the ACE blocking and the IL-6 receptor blocking work. Alex Kluzny and Anna and his team did all of the IHC work. They're my colleagues in the antibody development team on the IHC front. and Steve Orstad is our R&D Systems brand product manager who helped me with these slides. And then I just wanted to mention, please reach out to Lulu Koval if you have any questions about any of the products that I mentioned, if you have any need for any custom work at all that you might be interested in. She's your local technical service sales consultant, and she would be happy to answer any questions you have on product fronts. And with that, I will say thank you, and happy to take any questions.
All right, thank you so much, Jody, for such a wonderful talk. We really appreciate it. That was great. So I'm slightly curious for some of the ACE antibodies, of what fluorochrome availability is there available for these? So, as you're trying to build up some multicolor panels, would there be a wide variety of fluorochrome availability?
Right. So we have about 10 colors available. So we have a pretty good range of Alexa fluors. And then we have all the standard large molecules, the PEs and APCs and those types of colors.
Okay, great. Now, one of the things that I'm kind of curious about is different methods of antibody validation, for rigor and reproducibility, which is very important. So as you're talking about validation of antibodies and how there might be some challenges there, do you test for cross-reactivity of these antibodies to validate them to make sure there's no cross-reactivity across different protein variants?
We do as much as we can. So, for example, we try to guess what they might be. So, for example, if there's a protein in a protein family, we'll test the whole protein family to try to look for variability, in a cross-reactivity there. We can't test for everything, so it's we test as much as possible. So it's hard to know what it could be cross-reactive with. So we test as many we test as many members of the protein family as we have available. I guess that's the best way to say it.
Okay. Yeah, that sounds good. Just curious when talking about the antibody validation there.
Right.
And then do you know kind of the type of panel complexity that these antibodies have been used in? So typically what type of panels are people building up when they're starting to take a look at these ACE antibodies?
I don't think that anybody has really done that yet. So they've only been released for about a week. So I would love to see what people are doing with them, but I don't know yet.
You might need a little bit of time there. Okay.
Yep.
And then, I saw that you had the polyclonal antibody up there. And I'm curious, as opposed to the polyclonal, have you explored whether or not you might be able to use recombinant technology to be able to have some more specific antibodies as opposed to the polyclonal that might recognize different epitopes?
So like a recombinant, like try to figure out the epitopes of the polyclonal?
Yeah, so just, trying to utilize recombinant antibody technology to generate
sure.
Yep. As opposed to using polyclonals, which might recognize different epitopes.
Right. So we do have a recombinant antibody pipeline as well, and we do generate a lot of recombinant rabbit antibodies. So we do utilize that pipeline quite a bit. Most of these antibodies that I was talking about today were made quite a long time ago. So they were made in our more traditional hybridoma pipeline. And then the goat polyclonals, I mean, obviously those were made quite a while ago too. So these were things that we were kind of adding the application onto. But yeah, we do absolutely use recombinant rabbit development pipeline for new antibodies.
Okay, great. And then just a general question about, you know, antibodies and antibody titration.
Mhm.
Do you have a suggested, you know, concentration, you know, that you suggest for users to start off with when doing antibody titration, you know, to really make sure that they're using the appropriate concentration?
Um, and that definitely differs for each antibody. We have a range that we usually suggest on the data sheets for each antibody, and that's a good place to start. But then I really do suggest that people do their own titrations, because it's also going to vary depending on your cell type. So what may be a very low expression cell type might not work as well as something that an antibody concentration that you might do on something that's higher expressing. So we do have ranges that we suggest to start with, but I think that it's a really important step for everyone to start when they're working with an antibody for the first time, to do some careful titrations to make sure they're happy with the staining they're seeing.
Yes, I definitely absolutely agree with that. So that's good.
Okay.
So I think that's all the questions for right now. Jody, thank you so much for a really wonderful, wonderful talk.
Great, thank you.
You're very, very welcome.